I'd like to now, it does give me great pleasure to welcome our next keynote speaker, Dr. Mark Kiel, the founder and chief scientific officer of Genominom. Have I got that right, Mark? Genominom.
You nailed it. That was awesome.
Wonderful. Didn't even need to Google it. Mark completed his MD and his PhD and molecular genetic pathology fellowship at the University of Michigan, where his research focused on stem cell biology, genomic profiling of the hematopoietic malignancies. I got that one right?
Blood cancers, yep.
Brilliant, thank you. And also in clinical bioinformatics, I believe. He is the founder and CSO of Genominom, where he supervises the scientific direction of the Genominom suite of AI-driven software tools. Mark, it's wonderful to have you join us, and we are grateful for your time. The floor is yours. I do believe you have a presentation.
Yep, I'll just switch over the slides. And
Super.
While I do, if I can multitask, I just want to thank Priedemann for that presentation. That was a fantastic overview of challenges and opportunities in AI for pharma. For the purpose of my talk, if my screen can be seen, Roxy, is that the presentation mode?
I can see, yeah.
I'm going to take a deep dive into one of those specific aspects that Priedemann brought up, which is to say genomics. And I'm going to do that by way of example for one particular disease. Again, the example here being genomics as a great use case for how AI can be brought to life in the service of better treating and identifying patients with devastating disease, and ALS being one specific disease, though Genominom works across disease types, including cancers and rare disease. So the substance of my talk today, very brief, is an AI-driven locus-specific database for 35 ALS-associated genes, ALS being a neurodegenerative disease, and a locus-specific database being the variants that cause the disease in any one of those 35 different genes. So a bit of background about Genominom: the value that our company provides to our clients and our users leverages AI to make genomic information actionable. And we do this in the service of better understanding, better diagnosing, and better treating rare diseases and cancer patients. And on the pharmaceutical side, we use that information to create precision medicines targeted at molecular drivers of disease. Since we're talking technology here, this is one of the core technologies in Genominom's capability. We refer to it as genomic language processing, or GLP, which I may refer to later. I call this slide the order from chaos slide, where on the left is the chaos of the evidence in the published clinical and empirical or functional literature. In the middle there is what our GLP capability indexes out of that chaos: the diseases, the clinical phenotypes that the patients present with, the therapies that have been tried in patients or in animal models or in vitro, and the genes and their genetic variants. Importantly, no matter how an author describes any one of those entities. So not only does our GLP index that information from a broad corpus of that reference material, again, clinical and functional, but we also organize that information into what we refer to as genomic associations. How those genes and the variants in those genes associate with disease causation and what particular phenotypes are occasioned by them, and critical to what we're talking about here today, what therapies have been used to treat them or might be used to treat those different diseases. So I mentioned that GLP is one of our core competencies, and that's exemplified here on the left, which is AI-driven indexing of that data to put together that information into informative associations. The computational component that we bring to this challenge of understanding and making actionable this genomic information ensures maximal sensitivity, making sure that we're not missing anything. So every piece of that evidence is automatically indexed and organized into those associations. But the other flavor of our technology that is also AI-driven ensures maximal specificity of that data, which is a critical necessity when you're dealing with high throughput computational screening of information, such as AI techniques often afford, is to ensure that you've got the highest accuracy. And for that purpose, we have internal AI capabilities that allow our expert team of curators to expertly manually review that information to ensure maximal specificity, accuracy, and appropriateness to specific hypotheses for our pharma clients who have a particular drug development thesis, or otherwise to ensure that we're getting the right information that's idiosyncratic to particular diseases or genes. And for that purpose, we have an AI-driven computational queue or software suite that allows us to do that very rapidly and accurately at scale. And one manifestation of that I want to spend the rest of my talk talking about is in the service of understanding a complicated, clinically heterogeneous, genetically challenged disease, neurodegenerative disease called ALS. I won't spend too much time talking about the clinical features of ALS other than to say it's heterogeneous and complicated, but rather I'll focus on the AI capability that we have brought to bear to better understand this disease. It has a genetic component to it. That genetic component has to do with more than 36 different genes, any one of which, when mutated, can lead to disease. The landscape of information that we produce for each of those causative genes is a deep understanding of which genetic variants, genetic changes in those genes confer disease to those patients. Based on what evidence? All of which is curated to clinical standards to ensure the highest comprehensiveness and accuracy of that comprehensive causative genetic variant landscape for this devastating disease. So I'm showing some statistics there, but importantly, this is computationally organized for comprehensiveness and expertly curated for clinical grade accuracy. And we're able to do this for any given disease or set of diseases. We put that together in the landscape that I referred to, and there's two types of those landscapes that I want to exemplify here. One is the variant landscape, where the clients that we engage with have a need of understanding all of the variants for the purposes of drug discovery, target identification, or patient recruitment and enrollment, and ensuring that the right patients are enrolled in their trials. And I've mentioned before what this comprises, but I'll emphasize that it's every variant in any one of those genes ever published with all of the necessary evidence to support their interpretation and meaningfulness moving forward for drug development or clinical diagnosis and treatment. This is a summary of the data feeds that feed into and comprise our variant landscape. I've mentioned all of the published literature, the full text, the supplemental data, the figures and tables, etc. All of the clinical information and the functional information that comprises the literature component, exhaustively indexed through that computational GLP technique that I talked about. Coming along with that is the functional studies, in vitro, in vivo, in animal studies, etc., as well as the population frequencies of these variants, which suggests whether they're benign or potentially disease-causing, and then predictive models of pathogenicity or protein damage that might confer increased risk of clinical disease. All of that information put together is what comprises a variant landscape. And importantly, we have internal tools that fully automate the organization, annotation, and aggregation of that data. We have capability to manually, expertly review that information, and then we follow up that manual annotation and understanding of that data through an automated scoring that conforms to clinical grade frameworks. So the example that I want to give you is one of those 36 plus genes causing ALS, which is one of the main genetic drivers. It's the gene called SOD1. Just to give you a reflection of the power of this combined AI computational approach, is a previous background understanding through an engagement was of 70 causative variants in SOD1, whereas when we were asked to understand the full landscape, we increased that understanding by nearly 5-fold. And I'm exemplifying that here in a Venn diagram where the previous understanding may have come from a public database such as ClinVar, which is a collection of clinically submitted variants that were found in patients for this gene. When you look, though, at the data that we have assembled, which is shown in the crescent in green on the left, we have dramatically increased the understanding of those causative variants, which not only helps you better understand the nature of the variants that cause disease, but also increases your likelihood of identifying patients who are amenable to treatment, particularly when you have a precision therapeutic. This is a different way of looking at that same data now, where we're opposing those variants, which are shown here as circles, against the linear structure of the protein. So when you unfurl the protein from start to finish and then plot out where those variants appear in that protein and cause disease, what you're seeing here now at the top is the reflection of our comprehensive variant landscape compared to the very shallow understanding that's available absent a AI computational technique. A little bit deeper is, I mentioned that we understand the diseases, the phenotypes, the therapies for those patients. We also understand demographic information such as their age at onset, the disease severity, et cetera, and do so at a very granular variant level. And what you're seeing here is, again, that same protein structure with the variants here now in the different columns, and the circles being reflective of individual or groups of patients who have those particular variants, and on the y-axis, what their age at onset was. So if I focus your attention on the main message here is, some of these variants have a dramatically earlier age at onset for the development of ALS symptoms compared to others. And that's an important indicator from this computational data that we've expertly curated to say that not all variants in this gene are created equal, and some confer exaggerated risk to the patient, in this case in the form of earlier age at onset. I mentioned we also capture information that has to do with the rate of progression. So do you have an indolent course of disease that proceeds over 10 years? Or do you have a very rapid course of disease that takes place over a year or two before ultimate demise? And what you're seeing here is that some of the causative variants, based on the evidence in multiple patients who have that variant, confer a slow rate of progression of disease compared to others that confer a fast rate of progression. So again, this is just an example of the power of AI to organize this data, the power of AI to allow us to curate that information to maximal effect, and some of the types of insight that we can glean from that work. Here's a different way of looking at that data, now adding 2 additional genes, the FUS gene and the TBK1 gene, which number among the 3 dozen that cause ALS, where you see here now the age at onset is plotted along the x-axis, and the number of patients that have those different ages of onset are plotted on the y-axis in these distribution curves for each gene. So SOD1 on average confers a median age at onset of 45, whereas patients who have ALS because of FUS mutations have a mean age at onset that's a decade earlier, and TBK1 variant patients with ALS have a presentation that's 5-6 years later. So you're starting to see a distribution in the nature of this disease depending on which of the genes that drive disease are to blame. Lastly here, I'll exemplify our capability again to understand this information at a very deep level, computationally indexing for maximal sensitivity and completeness, and then expertly curating that information with our AI curation capability. This comes in the form of our patient landscapes, where now, before I talked about every variant ever described in any one of these genes, now I'm talking about literally every patient ever described in our entire corpus of clinical and empirical scientific literature. We know each of those patients, we know their genotypes, we know their ethnicities, their ages, other demographic and phenotypic information, as well as their response to therapy. This just diagrams those capabilities that we have of deeply understanding all of these patients and all of their particular presentations. Again, emphasize that this is computationally organized for scale and then expertly reviewed for accuracy, with all of that information, every scientific assertion being reviewed by a human curator. The example that I want to conclude with is now every patient with ALS who has a genetic lesion in the C9orf72 gene, the most frequently mutated gene causing ALS. We have put together a comprehensive patient landscape of more than 2,300 of these patients with all of those annotations that I talked about earlier, which culminates in a deep understanding of things like the nature of their clinical diagnosis and the probability of survival, which is displayed here in these different Kaplan-Meier survival curves, depending on the precise nature of their clinical diagnosis, whether it's ALS or motor neuron disease or frontotemporal dementia, say. Again, all at the hand of any one of those genes that I described. Lastly, here is a distribution of the patients and their ethnicities based on the number of reported cases among those 2,300 plus individuals that we've exhaustively curated for all of those different components that I had talked about. So again, I think Priedemann did a fantastic job of setting this up, where I wanted to take a very focused view on one aspect of AI, and that is in the service of better understanding genetics and genomics, and how that is used to understand the heterogeneity of disease causation and provide insight for pharma into how better to treat that, that genetic disease. So with that, I'll turn it back over to Roxy unless there are any questions.
Wonderful. Thank you so much, Mark. We do have several questions in the Q&A box, but I think we might save some of them towards the end, if that's okay with you. Wonderful. Thank you so much for such an enlightening presentation. I can see several comments and chats already that are engaging with a lot of what you spoke about, for sure.